Welcome back, students. In the first part of this video, I have already discussed about working principle and design of flow cytometry. Now, in the second part of this video, we will discuss data analysis and certain applications of flow cytometry. All of you know that by adding the fluorochrome to specific antibodies, it is possible to simultaneously detect multiple populations of cells and several cell types simultaneously. In flow cytometry, whenever a cell passes through the laser light, a voltage is generated. Fluorochrome is excited by this laser light and will emit photons, and photons will hit the photomultiplier tube, that is PMT. It is a detector and convert it into electrical signal. This electrical signal then travels to the amplifier and is converted to a voltage pulse. When cell starts entering into a laser light, voltage pulse is created like this. Now when it is inside or between the laser light, whole cell passes through the laser beam. So at this point, this is the highest voltage pulse. When cell leaves the laser light, the voltage pulse starts decreasing. For each pulse, height, width and area is measured. The number or size of the pulse depends upon the number of photons that are generated through the fluorochrome and is detected by the photomultiplier tube. Now, the data for each of these events is plotted independently and can be represented by different plots. You can see here this is a histogram. It is univariate plot, that is it allows the visualization of only one optical parameter, either fluorescence, light scattering. For example, here histogram displaying fluorescence intensity versus event number. So on x-axis it is fluorescent intensity and on y-axis it is number of cell population. As you can see here, the intensity of the plotted signals increases from left to right. That is, cell population to the left shows lower signal intensities than population to the right. Now, dot plot. Dot plot, it allows the visualization of two optical parameters in one graph. Hence, it is a bivariate plot. For example, in this dot plot, cells were analyzed using fluorescent conjugated antibodies or using FITC. FITC is fluorescein isothiocyanate. This fluorescein isothiocyanate is a derivative of fluorescein. Hence, the plot indicates cell population that are either negative for both the parameters or positive for a single parameter or positive for both the parameters. Another plot is density plot, which also depicts the distribution of cells within a population. In this plot, the red color represents the highest density of events within the cell population. With decreasing density, the color transition from yellow over green to blue. Another plot is contour plot. It is actually the extension of dot plot. It gives you a circularized pattern of signals. In the center, it shows highest signal. So in this way, the data can be analyzed with the help of all these plots. And the purpose of all these plots are to look for the individual property. For example, if you are looking at the single property varying in different types of cells, then you can use the histogram plot. But if you are looking for two different properties, then you have to select or you can use the dot plot. Is the selection of only a certain population of cells for analysis on a plot. That is, it allows the ability to look at parameters specific to only that subset. Here are different types of gating: rectangular gates, elliptical gates, polygon gates, quadrant gates, histogram regions. Earlier, when you start the reaction, first you have to do is side scattered versus forward scattered plot, and then you will get a cell population. These are the population of neutrophils or monocytes, whereas this is the population of RBCs. So what is gating is, if I have collected suppose 10,000 counts, then suppose I am only interested to see the population of RBC or B cells, then in that case in a dot plot you have to get a particular type of signal. So gate is it is actually electronic way of it is a selecting of a particular cell population, right? So this gate is a numerical or graphical boundary, as you can see here, elliptical gate. So this is a graphical boundary that can be used to define the characteristics of a particles to include for further analysis. So gating process is simply selecting particular area of interest on the scatter or histogram plot generated during the flow experiment that decides which cells you want to continue to analyze. Now you can see here, these are the most popular fluorescent dyes used in flow cytometer. All these fluorescent dyes have different emission wavelength, and that can be a huge event to label the sample with different dyes, because it creates a problem with different types of dyes, especially if you are doing two color labeling. Then you have to go through the process of compensation. So let us see what is this compensation problem. When the wavelength of two fluorochromes overlap, you can see here, when the wavelength of two fluorochromes overlap, the observed fluorescent signal detected by the flow cytometer may not be the actual signal displayed by the cell. Or else, in other words, you can say that the cells appears to possess a surface marker or phenotype that it does not actually have. So, this fluorescence interference can be corrected for by adjusting the measurement parameters of the flow cytometer. Either you can do it manually or automatically also. This correction is termed as compensation. In addition, this problem can be avoided by carefully selecting fluorochromes that do not overlap. These are the wide range of applications of flow cytometry like immunophenotyping, cell sorting, cell cycle analysis, apoptosis, cell proliferation. Even this flow cytometer can monitor intracellular calcium flux also. So here I want to take only one application, that is immunophenotyping. Nearly 70% of experiment in flow cytometry are immunophenotyping. So what is this immunophenotyping? It means characterization of cells based on their proteins by labeling these proteins with antibodies. And these specific antibodies are again labeled with fluorescent dyes and molecules. So here you can see from the blood sample we have collected this peripheral blood mononuclear cells. Then you have to stain with fluorescently labeled antibodies against this CD3, CD4, CD8, the 7 types of proteins. And then you have to put this sample into a flow cytometer. Now we are analyzing these 7 proteins at a time, and based on expression of these 7 proteins we'll be measuring or analyzing the type of cells. After staining, sample is placed in flow cytometer, but before going to the graph, just look at these CD markers. So based on these markers, as you can see here, if CD3 marker is there, then it shows the presence of T lymphocyte marker. CD3, CD4, CD8 shows T lymphocyte marker. CD19 shows the presence of B lymphocyte marker. Similarly, CD25, then CD14, CD33, they shows the monocyte, presence of monocyte marker. So based on these markers, let us see how to do flow cytometry. So these are the graphs. In first graph, in this first graph, on x-axis having forward scattered and on y-axis it is side scattered. We get 3 kinds of cells: lymphocytes, then monocytes, and then granulocytes. So first we have to select the lymphocytes, and these lymphocytes we are looking on this x-axis. It is CD3, which is labeled with Alexa fluoride, and on y-axis it is CD19. Now here, these are the lymphocytes. These are CD19 positive cells, means B lymphocytes. And these are the CD3 positive cells, means T lymphocytes. Now these T lymphocytes we can analyze further on y-axis. On this x-axis we are having CD4, and on y-axis we are having CD8 cells. So these are the CD4 plus T cells, means helper T cells, and these are the CD8 T cells, that is cytotoxic T cells. Now CD4 plus T cells, we can analyze it further. This CD4 T cells, we can analyze it further for two markers. One is CD4 and CD25. And here we can say these are the cells which are positive for both CD4 and CD25 as well. So these are our regulatory T cell. So from lymphocytes we enter, as you can see here from the first graph, from lymphocyte we enter into T and B cells. From T cells we enter into cytotoxic T cells and helper T cells, and from helper T cells we entered into regulatory T cells. Now you can also analyze that in a given sample how many regulatory T cells are present. So it's a very good application of immunophenotyping when you are having autoimmunity, or if you want to analyze stem cells, or you want to analyze differentiated cells, embryonic stem cell, you just need to have knowledge of specific proteins present on these cells. Use antibodies against those proteins and will get the exact number of how many cells are present in a particular sample of your interest. Here also here also we have selected monocytes, and these monocytes are analyzed here. So we have used two markers. One is CD33 and another is CD14. And these cells which are positive for both CD33 and CD14, these are called monocytes. So in this way you can do the immunophenotyping. Another interesting application of flow cytometry is analysis of apoptosis. This is also a very interesting application, and it is actually a pathway where the programmed cell death is occurring, and we can analyze it by flow cytometry. So the just understand the basic principle of this application. So we are having live cells or healthy cells. Inside these healthy cells, we are having the presence of phosphatidylserine, which is present inside the cell of plasma membrane. If a cell is entering an apoptosis, then membrane flips and the phosphatidylserine comes out. And we use the fluorescent labeled protein annexin, and annexin have the capability to bind to the phosphatidylserine, which is now flipped outside. So initially cells which are live and healthy cannot bind to annexin, and they cannot be stained by propidium iodide, because propidium iodide stain can enter only in dead cell. So our live and healthy cells are annexin 5 negative and propidium iodide negative, that is Pi. Once the cells are going in early apoptosis, this phosphatidylserine is coming out, and now you are having AV positive, that is annexin 5 positive cells, because annexin can bind to phosphatidylserine, which is present outside the cell membrane, but still the cells are propidium iodide negative. Why? Because propidium iodide can enter only and only in dead cells. Finally, in late apoptosis, here the propidium iodide will enter the cell, and the cell become annexin 5 positive, that is AV positive, and propidium iodide positive. So we are having three conditions: live and healthy cells, AV negative and Pi negative; early apoptotic cells, that is AV positive and Pi negative; and late apoptotic cells, AV positive and Pi positive. Now experimentally we are taking these we are taking these cells and we get this particular graph. So on x-axis again we have FITC labeled annexin 5, and on y-axis we have propidium iodide fluorescence. So these are the cells which are AV negative and PI negative. That means they are our healthy and live cells, right? These are the cells which are only AV positive and PI negative. They are early apoptotic cells, and these are the cells which are positive for both AV and PI. So they are dead because of apoptosis. Machine can calculate the percentage of all these cells. Now, if patients is treated with medicinal drugs, then this is the low concentration drug treatment, and we can see here this is increased cell in apoptosis and increased dead cells. And if you are increasing the concentration, then you are getting very high number of apoptotic cells as well as dead cell. So this kind of analysis of apoptosis by using flow cytometry can be used for anti-cancer drugs or cytotoxic drugs. It is very important application as far as pharmaceutical research is concerned. So thank you very much.